Hey guys, today we're going to talk about a few basic concepts with cells. We're going to talk about how cells were first discovered very briefly, and then we're going to talk about the difference between the two types of cells that exist on the planet, eukaryotic cells and prokaryotic cells. And then we're going to talk about the very first organelle, and that is the plasma membrane or the cell membrane. So let's begin. Basic concepts that you need to know right off the bat is that we are made of trillions of cells, and it is the cells that carry on all the processes that allow us to stay alive. Cells are responsible for the production of energy, the production of protein, reading DNA, reading RNA, the instructions for how our body does and can do and make anything that it can do or it can make. Now, organisms can be unicellular. They can also, such as bacteria. Unicellular organisms are just organisms that can carry out all of their functions by just simply being made of one cell. And then there's organisms such as animals, like we are, which multicellular, so they're composed of more than one cell, and those cells work together collectively to do a specific function. Now, look, there are hundreds of scientists that contributed to the discovery of cells, and I'm definitely not doing it a justice by just talking about two scientists. But I want you to pay attention to the year. 1665 was the year that Robert Hooke first took a look at these little boxes, as he called them. He observed these little boxes in cork. Okay, so he was looking at plants. He noticed that in this cork there was these little boxes, and it reminded him of the places that monks lived, and those were called cells. So that's where the name came from, and that was again observed in plant cells. Now, Anton von Leeuwenhoek in 1675, he was the first one to discover these living creatures that exist in pond water. Now, you guys looked at those in your lab that you did with the digital scopes when we looked at things like Paramecium and Euglena. Those organisms were some of the organisms that Anton von Leeuwenhoek looked at. And just wanted you to pay close attention to the name Robert Hooke, and remember that name. Also remember Leeuwenhoek, and remember the year. So this was like mid to late 17th century. Now, there's something called the cell theory that you absolutely have to be knowledgeable about, and this is just one of these things, guys, that you're going to have to commit to memory. So if you could, now, the diversity of cells that exist out there is just absolutely enormous. I mean, we have hundreds of different types of cells that exist in our body. And then when you look at the different species that exist on the planet, the diversity is just extraordinary. For example, the nerves in a giraffe's neck can be up to 2 meters long. So 2 meter sticks long. And you compare that to 0.2 micrometers long, like the bacteria. If you remember, you were looking at under 400 times magnification. You were looking at a bacteria culture that we got from yogurt, and it looked literally like these little tiny little colonies of lines that looked something like this, and that's under a 400 times magnification. So obviously we are in the micrometers when we talk about that. So a big difference in size, and the same thing can be said for the cells that make up our body. It's also important to notice that cells are actually three-dimensional, and when we look at them under a microscope, when we look at them on a piece of paper, we're always thinking of them as being two-dimensional, but they're really three-dimensional, and that's something that we have to keep in shape. The other thing is that the shape of the cell is limited in size by the ratio, and this is a very simple explanation. If you took all of the cells that make up your lungs and you were to spread them out, and you were to add up all the surface area that exists in your lungs, it would take up about a half a tennis court. So here's my tennis court. And half of this would be covered in your alveoli, or your lung cells, and that's a huge surface area to fit into the body of a person. And the reason that that has to happen like that is because we have to have tiny little cells so that the surface area is very high. Now when you think about it, the reason that we need a really high surface area is because we have to, and our cells need to, the plasma membrane, or the outer layer of our cells, needs to allow things to come in and things to go out. But mostly it's for absorption of things like oxygen and any kind of nutrients that we would need. So this is why we have to have a huge surface area in comparison to the volume. Otherwise we would have to be enormous creatures. And if we were enormous creatures, then we would have to use and expend tons of energy, which would make the demands for what we need to eat significantly higher. And we would put a bigger burden on the planet, and there would be fewer of us on the planet as well. All right, let's talk about the difference between eukaryotic cells versus prokaryotic cells. I think in my slide show that I talked about in the lab that we did, I talked a little bit about the difference between prokaryotic cells versus eukaryotic cells. You guys have got to remember that prokaryotic cells are ancient cells. Okay? And the reason that we call them ancient cells is because they were the first, or I'm just going to say they're the oldest cells. They've been around the longest period of time. They're very simple. Okay? This is a very simple cell. This cell doesn't have any membrane-bound organelles. Okay, it doesn't have any membrane-bound organelles. In fact, the DNA is just kind of floating around in here. It has some ribosomes that are floating around. It has a flagella, this long whip-like tail, which lets it move. It's got a capsule, so if it's in bad conditions, it can kind of stay dormant and survive until conditions improve. It has a cell wall, which gives it structure and strength, and it has a plasma membrane to control what goes into and what comes out of the cell. But that's basically it. So this isn't, we're not talking about something that's very complex. This is an old cell. It has, it lacks most of the organelles that you see in the eukaryotes. So the eukaryotic cells, these are you. Okay, these are the ones that are in you, and your body is made up of eukaryotic cells. All animals, all plants, all fungi, protists, these things are all made up of eukaryotic cells. So if we took all the cells in the world and we were to divide them into two categories, these are the two categories we would divide them into: eukaryotic versus prokaryotic. Now you guys are going to have to know all of these different organelles and what they do. You're going to have to know what the lysosome is and what it does, what the ribosomes are, the cell membrane, the smooth ER, all the parts of the nucleus, the rough ER, the mitochondria, the cytoplasm, the vesicles, and the Golgi apparatus. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different parts of the cell. And you have to know what their function is, and we're going to work on memorizing that, because I think that's the best way to tackle this. Now you're also going to need to know what a flagella is. You're going to need to know what cilia are. You're going to need to know what a cell wall is, chloroplast, central vacuole. So when you add it all up, there's probably about 20 different parts that you're going to need to know. Here's just a little bit more and a little bit of review in words on what we were talking about earlier. Internal organization, how a cell is organized. A eukaryotic cell contains a nucleus, and this is what directs all the cellular activities. It's the boss. The organelles, these are the parts, these are the employees. And they carry out all the functions the cell needs to do in order to survive. Eukaryotic cells, these are modern cells. These are much more advanced. They have membrane-bound organelles. They have lysosomes and the endoplasmic reticulum and the Golgi apparatus. Prokaryotic cells, these are the ancient cells. They have no nucleus and they have no membrane-bound organelles, but they do contain ribosomes. So remember, both of these contain DNA and both contain ribosomes. All right, let's talk about the plasma membrane for a few minutes, and then I'm going to talk about allowing things in and out of the plasma membrane, and then I'll conclude our lecture for the day. So the plasma membrane, every single cell has this outer layer. It's called the plasma membrane or the cell membrane. And this is what controls or allows certain things to come in and go out of the cell. And I think that a really good analogy is that the plasma membrane is actually a bouncer. Or it's a security guard, or it is a police officer or a border patrolman. This is something that allows, things that are supposed to be inside the cell in, and it prevents things that aren't supposed to be inside the cell out. And what this does is it contains proteins. They go all through the membrane, and they allow for a certain passage of materials. And these proteins are surrounded by a phospholipid bilayer. Notice that the word phospholipid bilayer, one thing that we should remember about lipids is that lipids are non-polar. Lipids are hydrophobic or water-fearing. This is what allows your cells to basically be waterproof. So the membrane is made predominantly of lipids and proteins. There's 4 main parts of the plasma membrane, and yes, you need to know these. There's the phospholipids, which makes up the majority of the protein. There's the glycolipids, which is used for cell recognition, so when cells are bumping into each other, they can recognize that they're not foreign. Cholesterol, which helps the cell actually maintain its strength. And then there's various proteins that are embedded inside of the plasma membrane that allows for cell transport to occur. Here's a picture of a plasma membrane or a cell membrane. And what you notice about this cell membrane is that these things right here, each one, this with these little tails and this one with these little tails, that's the phospholipid bilayer. So all this and all this and all this with their tails coming down here, that's a phospholipid bilayer. And embedded within the phospholipid bilayer can be things like cholesterol. Carbohydrates can be attached to these recognition proteins or these channel proteins that are embedded throughout. And all of these things work together to basically allow for certain things to move in and out of the cell. And I'm going to move ahead and then backwards. And when this got reformatted to fit this software, it kind of screwed this slide up a little bit. But let's just talk about 2 different types of transport. Basically, what this means is how things get into and out of the cell membrane or the plasma membrane. We're going to talk about active transport and passive transport. If something's active, that means that it's requiring energy. If something's passive, then it's not requiring energy. The same can be said for this. Active transport is describing how something can get into the cell with the help of energy or with the use of energy. Passive transport is showing how something can get into the cell without using energy. My best example that I can give is if I'm holding a book close to the ground and I want to get the book to touch the ceiling, I have to go against the law of gravity. So I have to add energy to make the book touch the ceiling. If I'm holding a book at the ceiling and I want it to touch the floor, I can passively do this. I can simply let go of the book. This will allow the book to go with the law of gravity and fall to the floor. The same can be said for things that are outside of the cell and want to come in. Let's start by talking about passive transport. There's three basic ways that things can passively, or without energy, move into the cell. The first one is called diffusion. That means that these are nonpolar substances that can simply pass through the cell membrane. This could happen because there's a higher concentration of these particles, whatever we're talking about. Let's say we're talking about, well, let's leave the specific examples out for now. But if there's a greater concentration of something outside of the cell than there is inside the cell, then what does nature want to do? Something that I've talked about before in this class is that nature wants to balance things out. So, if there's a higher concentration outside, then something's going to want to come inside. And the opposite is true as well. Facilitated diffusion is if we have something large, maybe something large or something that's polar, and it needs to get into the cell. It can do this through a protein channel. This is called facilitated diffusion. It means there's a big protein that is facilitating the movement of this large particle into the cell. But because it's coming into the cell, there must be a higher concentration of it outside the cell. Osmosis. This is the movement of water. And again, this can be said as a passive act of transport. If there is 90% water outside the cell and 80% water inside the cell, water is going to want to move into the cell. It will do this without any energy being added. We say that that is with the concentration gradient. And that's with the law of nature. It's kind of like something going with gravity. Active transport works very differently. This requires a pump, something that actively is adding energy. It's kind of like in a boat, there's something called a bilge pump. And when the boat takes on water, you have to push a switch on the bilge pump to pump the water out. The water getting pumped out, that pump actually uses electricity to pump it out. It's using energy to actively pump something out of the cell. Endo and exocytosis works the same way. Endocytosis is using a vesicle to move something large into the cell. The prefix endo, think of that as into. And then exocytosis is using a vesicle to move a large particle out of the cell. This can be done for polar or large molecules. Passive transport is going to be done with nonpolar or small particles. Hey guys, today we're going to talk about some of the distinguishing characteristics between animal and plant cells, as well as some of the organelles that exist within both of these types of cells. The first thing I need to remind you about is that both plant and animal cells are eukaryotic. This means that they contain cells or organelles that are nuclear bound. So what you should notice here is that all of these organelles that I'm circling, all of these organelles have what we call a nuclear envelope, or they are attached in a way that allows certain things to go into and out of. So they're basically separate compartments of specialized parts of the cell that do specific functions. This particular cell that we're looking at now is a plant cell. Plant cells are often distinguished by their square shape. In addition to their square shape, they're also characterized by their large central vacuole. The central vacuole allows for them to hold large quantities of water so that they can survive times of drought. In addition to a central vacuole, they also have these large green organelles which are called chloroplasts, and that's where photosynthesis takes place. So, in review, the main differences or main things a plant cell has that an animal cell does not. square-shaped cell, central vacuole, and chloroplasts. Other things that they have in common are the Golgi apparatus, peroxisomes, plasma membrane, cytoplasm, rough ER, nucleus, smooth ER, and so on. This is a picture of an animal cell. Notice that it is more round in shape. It lacks a flagella. It also lacks cilia. Now there are some types of animal cells that actually do have a flagella or cilia for movement. Parts of the animal cell that are noteworthy is the fact that it lacks a cell wall. It just has a plasma membrane. That's this outer layer here. So that's our plasma membrane or our cell membrane. It controls what goes into or what comes out of the cell. It also has a smooth endoplasmic reticulum as well as a rough endoplasmic reticulum. Rough ER is given its name because of these little blue dots that are located all over it, which are called ribosomes. This is a main area in which proteins are made in the cell. This large purple area in the center is called the nucleus, and this purple area in the middle of the nucleus is called the nucleolus. The nucleus is surrounded by something called a nuclear membrane and also contains holes called nuclear pores. The mitochondria are these football-shaped organelles that have these like squiggly little cristae is what they're called. These little these things that improve surface area so that the mitochondria can generate power or ATP better. The ribosomes are responsible for making proteins. The endoplasmic reticulum is responsible for transportation. The Golgi apparatus is this pink thing, and we'll talk more about that in a second. The lysosome is responsible for destroying things that aren't supposed to be there. It does what we call lysis, which means to split. All the fluid that fills the area in between all of these organelles, this gel-like substance, is called the cytoplasm. Alright, let's talk about each one of the individual organelles that you need to know, and I will go through them fast. Remember, you have a pause button, so use it. First thing, nuclear membrane. This surrounds the nucleus, and what it does is it allows things to go into and out of the nucleus, just like the cell membrane allows things to go into and out of the cell. It has numerous openings for nuclear traffic, so molecules that want to come in, they have to be able to fit through the pore in order to get there, and the nuclear membrane is composed of 2 layers. This particular organelle is found in both plants and animals. The chloroplasts. The chloroplasts were those oblong football-shaped looking things that was made up of all these discs that were stacked on top of each other within the organelle. This is found in plant cells only. This is where photosynthesis happens. It contains a pigment called chlorophyll, which is what gives plants their green color. The cytoskeleton is made of 2 parts. It's composed of something called microtubules. In addition to those microtubules, it's also made of something called microfilaments. What this cytoskeleton does is it supports the cell and provides it with shape. So it allows the cell to keep its shape, just like your skeleton allows you to keep your shape and to support you. This is found in both plants and animals. The endoplasmic reticulum, often abbreviated ER. The endoplasmic reticulum is this network of tubes that is fused to the nuclear membrane. And what this does is it has the ability to store molecules, it can separate and organize them, and it serves as the cell's transportation system. I think of the ER as a highway within the cell. The smooth ER, or the SER, is the part of the endoplasmic reticulum that does not contain ribosomes. So this is the part of the ER that does not receive proteins that are made from the ribosomes. The rough ER, or the RER, is the area where ribosomes are actually embedded in the surface. The reason we call it the rough ER is because when it's looked at under a microscope, it appears to be rough because of the ribosomes that are present. The Golgi apparatus. The Golgi apparatus is another one of these squiggly looking organelles that looks very similar to the endoplasmic reticulum. One of the big, easy distinguishing characteristics that separates the Golgi apparatus from the ER is that the ER is attached to the nucleus. The Golgi apparatus is not. This is considered to be the packaging plant. It's a membrane that's found near the nucleus but is not attached to the nucleus. It is composed of numerous layers forming a sac. This is found in both plants and animals. The lysosome, or the word lysis is in that, it means to split. This is the digestive center of the cell. It is responsible for digesting proteins, lipids, and carbohydrates. It transports the undigested material to the cell membrane so that it can go through what we call exocytosis in order to remove any kind of waste products from the cell. These vary in shape. It depends on what kind of cell we're talking about, on how large or how active the lysosome is. The cell breaks down if the lysosome explodes, and this is what I was talking about in class. If your cell identifies that there is a serious issue with it, it'll actually cause the lysosome to explode, which will cause the cell to be digested from the inside out. In essence, what happens is the cell breaks down. Some students like to think of it as exploding. It's more like decomposing or breaking down. This is found in both plants and animals. The mitochondria we're going to talk about often when we talk about cellular respiration and how your body makes energy. This is the second largest organelle. It has a unique genetic structure, and when you read about endosymbiotic relationship or endosymbiosis, you're going to read about how genetic structure has to do with the mitochondria. This is a double-layered membrane. It has these inner folds called cristae, which I had mentioned in the previous slide or several slides ago. What happens inside of the mitochondria is that energy is produced, energy in the form of what we call ATP or adenosine triphosphate. We'll talk more about that later. Another thing that happens in the mitochondria is that recycling and decomposing of proteins, fats, carbohydrates, and forms of urea. This is found in both plants and animals. It's important to note that plants carry out cellular respiration or make ATP just like animals do. The only difference is plants also carry out what we call photosynthesis. Another organelle that we can find in both plants and animals are vacuoles. And vacuoles are these membrane-bound, and what that means is they're surrounded by a phospholipid bilayer. They're membrane-bound sacs that are used for storage, for digestion, and waste removal. They contain a water solution, and there are other types of vacuoles called contractile vacuoles which actively pump water out of the cell. Vacuoles are important, very important for removal of waste, and also very important for storing things so your body can use them later. The plant has something called a central vacuole, which is just much larger and centrally located in the cell. Most central vacuoles in plants are easily observable under a microscope. Cell wall is only found in plants. This controls something called turgidity or turgidity. If something is turgid, what it means is that it is full of water, and so it allows it to stand up. If you've ever seen a plant that is wilted over, it's because there's not enough water inside of the cells. Once you add water, the plant then stands up. The plant stands up because the cell wall can actually be fully inflated, if you want to think of it that way, and it allows the plant to stand upright. Since plants don't have a skeleton, they rely on the cell wall to hold them upright. There are two different cell walls. The primary cell wall, which is really elastic, it has the ability to stretch very far, and the secondary cell wall, which forms around the primary wall after growth is completed. All I'm really interested in you guys knowing is that the fact that plants have cell walls, and those cell walls help the plant to stand upright. And that's where we'll stop for the day, so that gives you just an introduction onto organelles. I hope that you guys watched the video because this will help you significantly with the quiz that's coming up.